Thank you all for joining us today for the March seminar from the Western Center for Agricultural Health and Safety at UC Davis. I'm going to go over a few more housekeeping items and then we'll get started. Next slide. This seminar is being recorded both in English and Spanish, and you will be able to view it on YouTube in the next few days. All participants are muted. However, we welcome your comments and questions via the chat. We will have time at the end of the seminar to post questions to our speaker. As you have already heard, Spanish interpretation is available, and you can select the language of your choice using the interpretation icon. Closed captioning is also available. You can select the CC icon and click Show Subtitles. Next slide. For those of you who have not joined us before, thank you for joining us today. The Western Center for Agricultural Health and Safety has a mission to improve the health and safety of agricultural workers in the West through research, interactive trainings, and tailored outreach. Today, we get to hear from one of our own investigators and our deputy director. Next slide, please. Dr. Chris Simmons will be presenting on his biosolarization project, a soil health management technique and soil fumigation alternative that we believe has really important implications for worker safety. I am going to pass the mic and the screen over to you, Chris.
Great. Thank you so much for the introduction, and thanks for all of our attendees today. It's really a pleasure to talk to you about some of the work we've been doing within the Western Center. I'll just pull up my slides. So as you heard, I'll be talking a bit today about a process called biosolarization, which is really a soil management technique that can improve soil health across several dimensions, while also serving the critical role of disinfesting the soil and acting as a fumigation alternative. So what is biosolarization? At its heart, it's a way to harness passive solar heating and microbial processes within the soil in a way that makes that soil temporarily antagonistic to many soil-borne pests and pathogens, things like weed propagules, phytoparasites, microbial pathogens. And in this way, it can serve as a substitute for conventional pesticide-based control approaches. And one of the prominent ways that pesticides are used in California agriculture is to inject them or apply them through a drip system directly into the soil in a process called fumigation. Uh, and biosolarization can serve as a fumigation alternative. But we're also looking to go beyond the pest inactivation aspect of this process to really deliver multiple benefits to soil health in a way that you could never get with fumigation alone, to really increase the value proposition and create as many incentives as possible for a grower to potentially choose biosolarization as a fumigation alternative. So how is biosolarization enacted? Well, consider a plot of agricultural soil. What we would do is mulch that soil with a compatible organic matter soil amendment. So traditionally, this has been material like rice bran or wheat bran or mustard meal. As I'll talk about, there are many options for where you can source this material in California in particular, due to the wealth of agricultural products that are produced here. And so we typically will source a byproduct stream from some other food system in California. I'll talk about the use of almond hull and shells for this purpose today, as well as some other amendments. Once that amendment is applied, it's incorporated into the soil through disking or tillage, and then the soil is smoothed with a float or a roller. And then we apply drip line or drip tape and cover this whole setup with a clear plastic film. And in fact, the film that we use now is the very same tarp that you would use if you were fumigating your soil. Here it's been repurposed for a fumigation alternative. But once you have this amended, wetted soil beneath the hot California sun, that really activates this whole process. And so, to give you a glimpse of what's going on in the soil, we can think about the interface between the atmosphere, the tarp, and the soil itself. One thing that's happening is through this transparent plastic film, visible light from the sun is hitting the soil. It's being absorbed by the soil, and the soil is radiating out its own energy in the form of this longer wavelength infrared radiation. But the optical properties of the tarp are such that while it will let visible light through, it's more opaque or reflective to these longer wavelength infrared uh, light. And so that gets trapped beneath uh, the tarp. And in this way, we're promoting basically a greenhouse heating effect at the level of the soil, where you have this accumulation of solar energy driving an increase in heat and temperature within the soil. So this is the solarization part of biosolarization, where we're harnessing passive solar heating to achieve a temperature increase in the soil, and particularly in the valley in the summer, that heating in the upper levels of the soil can be rather extreme, on the order of 65 centigrade or 150 Fahrenheit plus. So in the topsoil, this heating effect alone can be enough to essentially pasteurize the soil and inactivate a broad spectrum of soil pests and pathogens. But recall that we do have that organic matter amendment present. And so the presence of that organic matter, the water, the heat, activates certain microbes within the soil. And what they do is they start to deconstruct, break down, and metabolize the nutrients in the amended organic matter. Now, the presence of the water filling in the pore spaces of the soil, the presence of the tarp on the surface of the soil blocking oxygen from getting into the soil creates an environment where this microbial activity is going to take the form of what we would call anaerobic fermentation. So these microbes are not able to exploit and use oxygen. And so instead of just producing carbon dioxide, they're going to produce a range of anaerobic fermentation products. And some of those products are chemicals that are classified as organic acids. These are going to be things like acetic acid, which you're probably familiar with in the form of vinegar. Vinegar is about a 5% solution of acetic acid. Other organic acids include things like propionic acid, which doesn't have a great odor. In fact, if you've had smelly socks or shoes, part of that odor is propionic acid. And then also compounds like butyric acid, which is something that you'll encounter in butter, even a component of your own stomach acid. So these are all chemicals that we're regularly or perhaps constantly in contact with. They're fairly innocuous in low concentrations, but they all share a property that they are natural or biologically derived pesticides. And so these compounds can be antagonistic to a broad range of soil pests and pathogens. Certainly, they have a much lower level of acute and chronic toxicity compared to conventional soil fumigants. So through the combined action of the solar heating, the oxygen depletion, the accumulation of these biological pesticides, you have this multimodal effect that's happening in the soil that can give rise to rather fast and robust inactivation of a range of soil pests and pathogens. So I want to talk about a example of how we uh, research and design a biosolarization process that's specific to a particular crop system. And here I'm going to use almonds as a major California crop that's very prominent in the Central Valley. And when you eat an almond, the part that you actually eat is the kernel of an almond fruit. So it's part of the pit of that almond fruit. And what surrounds that pit is a woody shell. And then surrounding that shell is a fleshy hull, basically the pulp of the almond fruit. And you can see some dried hull and shell here that gets removed in the process of isolating those kernels for edible almond production. So there's many, many tons of this material generated in California annually. Traditionally, it was used as uh, feed or bedding for livestock, but given the current market landscape for beef and dairy in California relative to almond production, there is a fraction of this material that is available for new recycling opportunities. And so we wanted to see if biosolarization could be a viable strategy to reclaim this material, essentially reinvest it back into agricultural soils. And in our case, we would take a circular economy approach and see if biosolarization with this material would be a effective approach to disinfest almond orchard soil. So this was a trial that we conducted in 2017 in Chico, California, in collaboration with a commercial grower. This was the Nicholas Nut Company. And what we have here is a picture of a site that was being prepared to house a future almond orchard. Before this photo was taken, for about 50 years, a walnut orchard had occupied this site, and over those 50 years, various infestations had accumulated in the soil. So it was a perfect test bed for what we wanted to do. It was a genuine agricultural context. We had a rather appreciable plot of land to work with, and so we could use all the same materials and operations that any other commercial grower would use to set up biosolarization. So what you can see here is our test site where you have the biosolarized rows with the plastic on them interspersed with these untreated negative control rows. And we can dig deeper into the specific treatments that we have here because there's a couple of different biosolarization conditions that we tested, uh, all using the almond hull and shell as the primary amendment, but different varieties of almonds are grown, and so you can get hull and shell from different varieties, and they have different properties, physical and chemical properties. The most popular variety of almond in California, and thus the world, is this variety called the nonpareil variety. Uh, has a very high kernel yield. Kernel looks very attractive. And so one of our biosolarization treatments was using the hull and shell from this nonpareil variety. But because almonds can't self-pollinate, you always grow multiple varieties together, such that between the three varieties, you can have basically a triangle of uh, pollination compatibility. So in addition to looking at non-Perel variety hull and shells, we also looked at biosolarization using hull and shell from some of these other pollinator variety of almonds. As a control, we had just completely untreated soil, so we didn't do anything to it. And then we also had a solarized control where we didn't amend the soil with anything, but we did lay the drip line, we did cover it with the tarp. So it had the solarization or the solar heating, but it was decoupled from the microbial processes in the soil so that we could determine how any effects might selectively relate to the solar heating alone or the solar heating in combination with the soil fermentation. So at this site, after we did the biosolarization, several months later, the trees were established. And so you can see the codes at the top there showing primarily NP or non-Perel variety trees in the rows, but interspersed with these other pollinator varieties, Monterey and Bennett Hickman. And since 2017, we've been tracking various measures of soil infestation and soil health and eventually crop health. So, looking at the initial changes to the soil, specifically the accumulation of those biopesticides, these organic acids that we know can knock out various pests and pathogens, what we see is that within the first 9 days of the 6-week biosolarization treatment, we see the greatest accumulation of these organic acids. So, most of the action in terms of disinfestation is probably happening within the first 10 days. And we see that acetic acid is the most prominent organic acid. So it could be doing much of the heavy lifting for that disinfestation. And that's a trend that we usually see across many different types of biosolarization conditions. And then in tandem, we looked at the control of various existing infestations. And one of the infestations at the site was of something called root lesion nematode or Pratylenchus vulnus. This is a plant parasite. So nematodes are small worm-like creatures, and in this case, they can form these lesions on the surface of roots and inhibit the plant's ability to effectively draw water and nutrients, can cause stunting or even death of the tree. And what we see here is that for our biosolarized and solarized treatments, again, by day 9, when we see that spike in biopesticide production, we see a very large inactivation or reduction in the nematode viability. And if we zoom in here, looking at day 9, you can see that there's a very small amount of live nematodes that hung on in the solarized treatment, and essentially we had a complete kill in the two biosolarization treatments. And another infestation at the site was of another parasitic nematode called ring nematode that also can colonize roots and inhibit nutrient and water uptake. And here we see again by day 9, the solarization and biosolarization treatments had completely inactivated those nematodes. And then in the subsequent weeks, we saw just a die-off across the board, and we think that that could be due to some of the tillage and other processes that were happening after our treatment completed. Probably was traumatic for those nematodes and caused the remainder of them to die off. But looking out further in time, so this is 2.5 years after that initial biosolarization, what we see is while a notable infestation persisted in the untreated control soils, for our biosolarized and solarized soils, we're still seeing a suppression of those root lesion nematodes even 2.5 years after treatment. And this would be comparable to the level of control that you could expect with conventional fumigation. So I mentioned that this is not just a disinfestation technique, it's also a soil health improvement technique. So we were looking at other metrics of soil health, including the presence and enrichment of fertilizer compounds. So nitrogen is one major plant nutrient called a macronutrient for plants. And almond hulls and shells contain protein, and so they contain some amount of nitrogen. And what we see here is for the biosolarized soils containing those hulls and shells, there's a multi-year elevation in the level of nitrogen in the soil. You can kind of think of the hull and shell as like a slow release fertilizer, where as they break down over time, microbes will take that organic nitrogen from the proteins in the hull and shell, mobilize it in their own biomass, and then as those microbes die off, they make that nitrogen available for the crop. And this same type of trend was seen for potassium. Potassium is another fertilizer, another macronutrient for plants. And because the hulls contain appreciable potassium, again, for multiple years after treatment, we see this enrichment in soil potassium, releasing those nutrients over time for the trees. And then just looking at overall soil carbon. So plants don't get their carbon from the soil primarily, they get it from the air. But what this soil carbon represents is kind of the enrichment and persistence of organic matter in general in the soil. And one of the key pillars of a healthy soil is a uh, certain level of organic matter within the soil, which does a few important things. It can help to kind of bind or glue the soil together in a way that allows it to resist erosion, but still allows air and water to readily percolate through it. It can allow the retention of certain nutrients within the root zone. It gives a substrate for microbes, a diverse set of microbes to persist in the soil. And it can also just represent carbon that's being sequestered in the soil that's not going back into the atmosphere and contributing to greenhouse gas emissions. So here we see, again, over about 2 years, a significant elevation in soil carbon and thus soil organic matter in response to that amendment with almond hull and shell. And especially in California, this can be very useful with regards to irrigation because organic matter is like a sponge. It can hold 3, 4 times its own weight in water. So if you're increasing the soil organic matter by 1 or 2%, that can have a tremendous impact on that soil's ability to capture and hold water in the root zone. And then going beyond the soil to look at the growth behavior of the almond trees in the different biosolarized plots. One interesting thing is that in the first year after the trees were planted, we didn't really see any benefit to their growth. And in fact, in some cases, the trees in the biosolarized plots were growing slower than those in the untreated controls. But that changed about 1 year after planting, where we saw that relationship flip. And that's when the trees in the biosolarized plots started to show a more rapid growth rate, as indicated by the change in their trunk diameter over time. And so, what this told us is that it does take some time for the trees to acclimate to the rather dramatic change in the soil physical and chemical environment coming out of biosolarization. But once they do adapt, you start to see the benefits of that soil treatment. And again, this was most apparent 1 year after biosolarization. So if we look at just over 4 years after those trees were planted, what you can see here is a uh, differential response to biosolarization by the type of almond tree or variety of almond tree that's being grown. Uh, so for the nonpareil variety of almond trees grown in the biosolarized soils using nonpareil hull and shell, that's where we saw the greatest benefit to growth rate. So too, for the Monterey variety, they responded most strongly to growth in soils biosolarized with the nonpareil hull and shell. For the Bennett Hickman variety of almond, we didn't really see any significant difference between growth in the biosolarized versus the control soil. So it does show that there's some interaction at play between how you biosolarize the soil and the type of tree that you plant. And so you could strategically choose varieties of trees that are going to be best primed to benefit from biosolarization. Another thing that we looked at was the health of the canopy. So this was done with aerial imaging, where either a plane or a drone flies back and forth and scans the canopy using different wavelengths of light. And based on differential absorption for these various wavelengths of light, you can calculate absorption indices that speak to the level of chlorophyll or the level of stress in the canopy as an additional measure of either health or stress in the orchard. And so here's just a snapshot showing how the aerial imaging at one wavelength mapped to that arrangement of treatments in the rows in the orchard. And we did this for uh, several seasons. And there were very few consistent trends here, at different time points. Occasionally, there would be significant differences between one of the biosolarization treatments and the untreated control. Again, it was sporadic, but anytime we did see a difference, a significant difference, it was always to suggest that there was some benefit to the canopy health in one or more of the biosolarization treatments. And usually this was via absorption indices that spoke to the level of greenness in the canopy, which could be affected by just the number of leaves, the amount of vegetation there, or the concentration of chlorophyll in the canopy. And so the thought is that if trees in certain seasons and certain treatments are exhibiting more chlorophyll when they're grown in these biosolarized soils, they're better able to capture sunlight, better able to capture and harness that energy. And the hope is that they would then be able to invest some of that energy into fruit production and ultimately almond yield. So almond yield was something that we were able to eventually measure in summer 2023 after COVID delayed our ability to get back into that field and do these measurements for a couple years. And what we saw here is that the yield benefits differed by the variety of almond grown. And what you can see in the upper right here is that for the nonpareil and Bennett Hickman tree varieties, there's a more subtle increase in yield when grown in the biosolarized soils. But for Monterey, that difference is more pronounced. And in fact, there was roughly a 20% increase in yield for that particular variety. What we see in the lower right is just a plot that's comparing the yield for any type of tree in any type of biosolarized soil to the yield of any type of tree in a control soil. This is basically looking at the global effect of any type of biosolarization on yield. And what we see here is that there was a significant overall global effect on yield for biosolarization, and it was about 4.5% average increase of yield across all the varieties and treatments. So this was really an exciting metric for us because it speaks to what is essentially the most important metric for growers. If you're going to invest money, time, and resources to treat your field in this way, does it deliver a measurable yield benefit, which would then allow for the process to pay for itself if it's generating additional income? And the answer here does suggest that is the case. So I also want to switch gears and show a different type of biosolarization study in a totally different region with a totally different to the Western Center's overall goal of enhancing pesticide use safety and mitigating exposure in the Western region. But here we have a study that's done in Arizona using the byproducts of date processing as a biosolarization amendment. So in this particular study, we looked at um, incorporation of dates, fruit that had been culled because they didn't meet a quality standard, used that as a biosolarization soil amendment, and looked at the inactivation of fungal pathogens in the soil as well as the growth of lettuce in the soil. So again, this was done in Yuma, Arizona, so very close to the California border and having low desert conditions that are relevant to California agriculture as well. So what we saw in this study, looking at a few different forms of biosolarization, um, and in this study, we were looking at either a wet or a dry condition, so fully irrigated through the drip lines as we would normally do, or minimal water application, really just the water that was carried over on the date paste mixture itself. And what you can see here are data that speak to the amount of Fusarium oxysporum inactivation in the soil. So this is a fungal pathogen that affects many different types of crops. And you can see uh, for the biosolarized wet condition, we achieve an over 3 log or 99.9% kill of those pathogens, and that's mitigated some amount by using a dry version of biosolarization. So your efficacy takes a hit in that version. And then comparing that to solarization, so the solar heating with no amendment in the soil. Uh, in this case, given the high temperatures of Yuma, Arizona in the summer, the solarization also did a very good job in activating fusarium, but it was completely ineffective in a dry version that lacked water. So it does show that the amendments of biosolarization can help to achieve some partial efficacy in a low water deployment, but really that water addition is key to maximizing the efficacy of biosolarization. It helps to support the microbial activity. The water helps to darken the soil and promote more absorption of solar radiation. And then that water allows that heat to conduct deeper into the soil as well. We did 2 plantings of lettuce in these treated plots. And initially, the first planting of lettuce did not show a initial benefit in terms of seedling survival. And in fact, there was a bit of a depression in seedling survival for those biosolarized treatments. So this highlights the importance of building in a remediation period between when you end biosolarization and when you introduce the plants. And so usually that period, depending on the conditions of the soil and weather and climate, could be anywhere from 2 to 6 weeks that you would need to let the soil sit fallow to really make sure that all of those natural biological pesticides have dissipated and are not going to affect your crop. Um, but here we see in the second planting of lettuce done several months after, we don't see any indication that there's any lingering toxicity in the soil. And in fact, for that uh, second planting, we see that there's a significant decrease in plants that show symptoms of fusarium wilt or fusarium disease, and a corresponding significant increase in the yield of the lettuce, specifically in that biosolarized treatment. And here's just a photo that shows lettuce in each of these different biosolarized or untreated control plots. And you can see the difference in the stunting and the wilting between the biosolarized and the control treatments. And then another interesting thing is when you amend the soil with this date paste, the dates have a purplish hue. And you can see in the biosolarized photo, the soil itself takes on that purple color. So it's a very striking, almost extraterrestrial appearance to have the green lettuce growing in this purple soil that you've biosolarized. So I've talked a bit about some of the overall effects of biosolarization and showing that it can work across multiple different pest targets and for different cropping systems. But we're also interested in understanding how and why it's able to do this. So here's a study that looks at some of the more mechanistic aspects behind biosolarization, asking, what are the volatile compounds that we're making in the soil during this process that could play a part in pest inactivation, but also we're interested in this question because we want to know what folks could be exposed to if they're working in and around these fields or if they're living in communities proximal to biosolarization sites. So in this study, we set up several biosolarization plots using different almond hull and shell amendments. And what we did is take samples of the soil at various time points and run them through what's called a solid phase microextraction process that basically allows us to heat up the soil and capture anything that volatilizes and comes off the soil for analysis and gas chromatography with mass spectrometry. So we can capture the volatile compounds that escape the soil, and then we can characterize them to figure out what they are. So here are some of the untargeted volatile profiles for each of these conditions. So looking at soil that was biosolarized with either no amendment or with these different varieties of almond hull and shell with either solar heating or just ambient temperature. And what you'll see here is that the application of the solar heating really plays a strong part in determining what that profile of volatile compounds is, because when you have ambient temperature, you're dealing with mostly terpenes, but when you apply that solar heating, then you shift to being dominated by volatile fatty acids. So here's a chart that shows some of the most prominent volatile compounds that were detected across our biosolarization treatments, and they fall into two categories. The volatile fatty acids, of which I discussed a few of them earlier, the acetic, butyric acids, and other longer chain volatile fatty acids. But then there's also these compounds that are called ketones. And in certain conditions, they can even be the most prominent volatile compound. So something like acetone, for instance, which is present in some food products. So we're also exposed to that. It has a very buttery flavor that's used in popcorn sometimes. So there's an overall trend here that many of these compounds have exposure limits that are much higher than conventional pesticides. These are compounds that do not have any known carcinogenicity at the concentrations that we detect them or just at all. And that's in contrast to some soil fumigants. So overall, we are elucidating the compounds that in tandem may deliver that robust biopesticidal effect, but also providing assurance that the specific compounds and the levels that we're measuring them at are not expected to create a hazard for workers or communities proximal to these sites, certainly not in comparison to some of the published exposure limit data or animal study toxicity data that you would have for conventional fumigants. And I wanted to show how biosolarization can promote safety in other contexts. So here we're using a completely different soil amendment. So this is grape pumice. This is the skins and seeds that are left over from winemaking. And we were looking to see if biosolarization could go beyond controlling plant pathogens to also inactivating human foodborne pathogens that can have a reservoir in the soil. So it can sometimes be that an animal walking through a field and it's manure or contaminated water, or in some cases, contamination brought by personnel in the field can lead to pathogens in the soil that then are carried over onto a crop. And so we wanted to see if biosolarization could disinfest some of these uh, ultimately foodborne pathogens. And in this case, we were targeting E. coli 0157H7 using an attenuated uh, strain of it that was not infectious but still had the same growth and inactivation behavior as the genuine pathogen. So here we were doing a small bioreactor study to simulate field conditions during biosolarization, where we could load these bioreactors with our amended soil and control the temperature, create an anaerobic condition, just as we would have in the field. And in this case, we were looking at two different levels of grape pumice, comparing that to tomato pumice, which are the skins and seeds from industrial tomato processing, and then complementing that with some compost and certain treatments to give basically the soil a kick of microbes that are very robust and able to break down a variety of organic matter. And then we took extracts from the soil and spiked it into liquid cultures of this E. coli pathogen surrogate and looked to see if it affected the growth of the pathogen surrogate over 24 hours. So what we see here are some of the optical density or level of growth data for those bacterial cultures in response to exposure to these different soil extracts. And what you'll see here is kind of this valley in the middle where there's significantly decreased growth of the E. coli in response to the grape pumice uniquely. So this is probably an instance of where the grape pumice has some natural antimicrobial compounds uh, like anthocyanins that give it that rich purple color. And those are simply leaching out during biosolarization, being complemented by other stresses like the soil heating, and are delivering an activation of, in this case, a human pathogen. So it may suggest that biosolarization can also be used not just for crop safety, but for food safety to help disinfest soils of human pathogens should there be a suspected contamination. And then also thinking about other aspects in terms of the mechanics and which microbes are doing the heavy lifting during biosolarization. So a few years back, we had taken sequencing data from a number of biosolarization studies that we had done over the years, doing a special type of sequencing that basically looks for the microbial fingerprints that tell us which species or which taxa of microbes are present in the soil. And we were trying to see if over all of the different studies and biosolarization conditions that we had done over the years, were there consistent trends in terms of which types of microbes flourished in the soil during biosolarization? What you see here is a network diagram where each dot represents a type of bacteria or fungus. Um, and when you have these colored clusters, what it basically represents is a group of bacteria and fungi that rise and fall together within the soil. So when one grows, they all tend to grow. When one starts to die off, they all start to die off. Um, and so we can think of these communities within the soil that are tolerant to the same conditions, and maybe they're performing some similar functions within the soil. One function that we care about is which microbes are likely to be producing those biological pesticides that are so important to that pest inactivation effect. And what we see here is a correlation between the level of organic acid biopesticides that accumulate in the soil versus the cluster in this diagram. So there are 8 different colored clusters here representing groups of bacteria and fungi. And what this diagram is showing is that cluster 2 is really the one that most consistently correlates to the level of organic acid biopesticides in the soil. So more than likely, it's microbes within that particular cluster that are making those biopesticides. And so cluster 2 here is the green cluster that you see on the right. And there are several genera that are within that cluster prominently. These are going to be bacteria like Acetobacter, Clostridium, Enterococcus, and some of these are known organic acid producers. So now we start to zero in on specific types of bacteria that can promote effective biosolarization because they can tolerate the harsh conditions of the soil during that process. They can break down a variety of organic matter amendments, and they can produce those crucial biopesticides that are necessary for the process to work maximally. So this is getting towards one of our aims, which is defining a minimum critical microbial community that either you can look for in the soil to make sure it's compatible with biosolarization, or you can introduce to the soil as an inoculum to make sure that you have all the microbes necessary for this process to work. So across these projects, we've had a number of collaborators, both within UC Davis and outside of campus. Many folks in my lab over the years have been part of these various projects, and I can't say enough about our industry collaborators as well. That's such an important connection to show that what we do in the lab can translate to an authentic commercial field context. So I want to thank everybody that we've worked with over the years, as well as our sponsors. Most of this funding has come through the Western Center for Ag Health and Safety and its support through NIOSH. Much of the work in the first half of the presentation was also supported through complementary funding by the Almond Board of California. And then for the work in Arizona, uh, that was supported through a generous gift from the California Date Commission, as well as additional support from the Western Center and USDA. And I want to thank our collaborators at the University of Arizona for helping make that project a reality. And I always like to end on this slide. What it is a truck showing all the lettuce from our field trial going to the Yuma Food Bank so that the good data would lead to a good meal for a few folks. And with that, I want to thank you all for your time and attention today. And if you have any questions, I'm very happy to answer them.
Wonderful. Thank you so much, Chris. I always enjoy your presentations. Let's see. So if anyone has any questions, please put them in the chat. I will get us started with so you did some trials in California, some in Arizona. What is the feasibility of using biosolarization basically in all ag settings where traditional fumigation is currently used?
Yeah, it's a great question. I mean, we've started with the Central Valley because the weather and climate work in our favor with the hot summers that really promote strong solar heating, and that can do a lot of the work to disinfest the soil on its own. But then there's this whole question of, well, there's plenty of fumigants used in the coastal production regions as well, particularly for strawberries and other berries. Can we make this process work there where we won't necessarily have the benefit of daily high temperatures of 110 degrees plus? And so we actually have a DPR grant right now that's aimed at that question to specifically develop biosolarization approaches that are geared towards the more moderate temperatures that you would experience in the coastal production regions. So in those cases, you're going to be leaning a lot more on the other stresses that you can induce in the soil. So any endogenous biopesticides that may already be in your organic matter amendments, as well as making sure that even though that the heating that you're able to achieve may not be lethal on its own, it can still be enough to support the fermentation that can give rise to these other stresses like oxygen depletion and these fermentation products that are pesticidal that could still get the job done, even though it may take a little bit longer because the kinetics are slower in the cool temperatures. So uh, those studies are ongoing right now. We're not the only group working in this area, and you can find some published studies that have shown for rice bran and some other conventional amendments that there are cases where it has worked very well in strawberries. We're trying to make sure that it's able to achieve those results consistently and across all of the coastal production regions. So there's definitely more to come on that front soon.
Great, thank you. So there's the weather component, but then there's also the supply of the amendments. Do you have any risk of running out of suitable amendments that you can use in the process?
We're seeing that, especially in California, there are so many byproduct streams that you can choose from. There's not going to be one material that's going to satisfy all the acreage that could be biosolarized. There's not enough rice bran to cover all the currently fumigated acres. There's not enough almond whole and shell or tomato pumice. But if you have a real bioeconomy where you can source these different materials that are co-located with your particular field, then you can have this patchwork of different materials that you might be able to draw from. And what we're trying to do is shine a light on those materials that could be compatible. And we consistently see that there are ways to make almost every type of labile organic matter side stream work. So we've looked at spent grains from brewing. We've looked at the cuttings from onion processing. Uh, we've looked at some of the more wastewater or effluent streams from different food processing operations. And as long as you have some amount of bioavailable carbon and nitrogen, it's usually enough to support fermentation to a degree where you can get this to work. It's usually a matter of how long you need to apply it or how much of this material you need to add to the soil. So uh, there are colleagues within UC Davis that are working to develop basically a digital atlas that will show at different seasons and different locations, here are the crop residues that are available that could be used for biosolarization or any other number of types of bioprocessing.
Very cool. Oftentimes in safety, when we think about changing out a procedure or a material or process, there's the opportunity to remove risk, but also introduce new risk. Do you see any new safety concerns that might emerge as people consider switching to biosolarization?
I think there's a lot more work to be done to fully characterize all the volatile compounds that could be produced during biosolarization and what level of exposure might you see in practice. The data to date aren't showing any clear indicator of increased risk or even comparable risk to what you would have for conventional pesticides. Um, but there does need to be a formal exposure and safety analysis, like you would do with any conventional pesticide. We're also cognizant of other risks that are outside of, let's say, the traditional volatile exposures. I mentioned that in its current form, for biosolarization, we're co-opting the tarp that's used for fumigation. That's more robust than we need because we're not trying to trap and, prevent escape of highly toxic compounds. Under its current form, we do have concerns about, well, are there persistent microplastics that could be introduced to the soil through the use of this tarp? So there's a student in my lab who's looking at the use of bioplastics as an alternative to conventional plastics, where you would not expect the same persistence of microplastics because they're biodegradable materials. So she's looking at the fate of bioplastic when used for biosolarization and confirming that these plastics can be as effective as the traditional fossil fuel derived plastics.
Speaking of the plastics and the way in which the materials are laid, someone's asking if you could walk us through the process of laying down the plastic and just kind of the equipment used. What does it look like in the field?
Yeah, we're very deliberate in trying to co-opt as many traditional agricultural operations as we can so that this process is as accessible as possible. So it's not going to be, exotic and use equipment and practices that growers are unfamiliar with. So we actually use the same tractor implement that's used to lay the plastic down for fumigation, which is basically a tractor drawn spool that will in one pass lay the tarp and also bury the edges of the tarp to create that seal. Um, so any operation that's currently fumigating could use that same implement to lay the plastic. In very small deployments, we've done it manually. That's also an option. Um, and that's also true for other aspects of biosolarization. So in terms of how we spread the amendment, that's the same type of spreader you would use if you were applying compost to the field. We use the same tractor drawn drip line applicator that you would use if you were applying that in any other context. So there's actually very little in the whole setup that's very, unique to something else that a grower might be doing anyways. And that's a theme that we try to keep so that nothing will strike the grower as being burdensome or outside of what they're already familiar with.
Right? It probably makes adoption easier to imagine.
Yeah.
Okay, so we talked about this a little bit before we got started. DPR is developing a sustainable pest management plan and is looking to identify some of the priority pesticides. How do you see biosolarization fitting into that equation?
Yeah. Well, that list has not been published yet, but I think a lot of folks do expect to see some fumigants pop up on that list, which would mean that they're targets for at least a 90% phase out over the next couple decades. So if that should come to pass, there will be a strong incentive to explore fumigation alternatives, and we want to make sure that biosolarization is positioned to be a viable alternative for growers, whether they're seeking an alternative to fumigation because of regulatory action or just because they're interested in some of the secondary soil health benefits that you might be able to achieve. Um, and what goes into that is basically generating as much actionable data as we can. Some of the most compelling data are going to be those that show the success of the technology in the environments where it's to be used with the same cropping systems that that grower may be dealing with currently. Now, we're not going to be able to hit all those marks alone. So thankfully, there are other groups in California and beyond that are also studying this. Uh, but ultimately, you need to have sufficient data to really remove the risk first and foremost and to make a compelling case that you'll see efficacy within a given context, which there are infinite contexts in terms of weather climate, soil texture, pest profiles, crop sensitivities, agricultural practices. There are a lot of variables at play here. Um, but we're trying to create as many published precedents as we can. And I'll say that my group is very open to talking one-on-one with any growers that may be interested in terms of how might you set up a small test plot in your operation to make sure that this process is going to work under your particular conditions.
Well, you read my mind because I was going to ask you how people could contact you if they had questions or wanted to learn more, perhaps about implementing this on their property.
Yeah, you can email me directly at cwsimmons@ucdavis.edu. You can also find my contact information through the Western Center site, aghealth.ucdavis.edu. Always happy to have that conversation.
Wonderful. Well, thank you so much, Dr. Simmons. We're really happy to have you present. We're so proud of all of the research happening here at the Western Center. Thank you all for joining us today. We will be back here again at the beginning of April talking about farm worker housing. So please join us. Thanks a lot. Bye.
Thanks so much.